Hello friends, dear learners and my dear student. First of all, I take an opportunity to welcome you on the lecture Ultraviolet Visible Spectrophotometry. Student, I am Dr. S.R. Labhade, working as Associate Professor in the Department of Chemistry. Student, in previous lecture we discussed laws of photometric analysis and we solved the problems based on the laws of photometric analysis. In today's lecture, we discuss the instrumentation of spectrophotometry, that is the instrumentation of spectroscopy. If we consider instrumentation of spectroscopy, the spectroscopy which is employed in spectroscopy employed in ultraviolet, visible and infrared region are called as optical spectroscopy. That means region, ultraviolet region, visible region and infrared region. Measurement of analyte in this region that is related with optical spectroscopy. Here, optical spectroscopy may be an absorption spectroscopy or it may be a emission spectroscopy. Absorption or emission spectroscopy. Therefore, in this spectroscopic technique, the components which are involved or utilized in optical spectroscopy, that may be absorption spectroscopy or emission spectroscopy, are similar type. And these components are, number one is a radiation source. Number one is radiation source. There is no spectroscopic technique without radiation source. Radiation source is the first important component of all optical spectroscopy. Radiation, that is actually the source which is utilized in this spectroscopic analysis. This source must be stable, it gives more intense radiation for the measurement. Second component, this radiation source emits all possible wavelength and therefore second component is a wavelength selector. Wavelength selector. It is also called as monochromator. The monochromator in absorption spectroscopy may be filters, sometimes prism or sometimes diffractive grating. The function of the monochromator is to isolate a monochromatic radiation. Generally, spectroscopic techniques are performed by using a monochromatic radiation. Third component is a one or more sample cell. Sample cell means a cuvette that goes the sample in light path. Sometimes in a double beam instrument we require two sample cells, one for sample, another for reference or blank solution. Therefore, there are provision in the instrument to put two sample cells and single beam instrument utilizes only one sample cell. Next component, which is converts radiant energy into electrical energy, and that is called as detector. Therefore, in spectroscopy we are measuring the degrees in radiant power or radiant power of specific wavelength. Therefore, here we have to detect this radiant power using some device. And device which is used for this purpose is a detector. Detector is spectroscopy that converts radiant energy into electrical energy and that signal feeds to the recorder. Therefore, fifth component is the recorder. Therefore, recorder that may be sometime digital recorder or sometime a data processor. In modern instrument, recorder is a computer system. Therefore, all these are the 5 important components of absorption and emission spectroscopy. Based on the different types of technique of analysis, these components are arranged differently in an instrument. Therefore, here the arrangement of the instrument, number 1, for absorption spectroscopy. The instruments are arranged in this type, that is a radiation source, then a monochromator, then a sample cell, then a detector, and finally the recorder. Therefore, in this case, all components are arranged in a single bin. Because absorption spectroscopy measures the decrease in radiant power of the incident beam, therefore absorbance is measured in terms of decrease in radiant power of incident beam. Therefore, here source, monochromator, sample cell, detector and recorder all are placed in a single line or in a single axis. Therefore, here decrease in radiant power that can be easily measured in absorption spectroscopy. Absorption spectroscopy may be a molecular absorption spectroscopy or sometimes a atomic absorption spectroscopy, but the arrangement that will be same. Second type, that is called as emission spectroscopy. In emission spectroscopy, the radiant power emitted by the sample is measured. Therefore, emission spectroscopy, the arrangement of the sample component, arrangement of the instrumental component are little bit different. Therefore, here there is a first source. From this source, a monochromatic radiations are selected, monochromator. Then here a sample, then again second monochromator, then a detector or sometime that may be a recorder. A recorder may be not in the line of detector, it may be away from the detector because electrical signal that is passed by using an electric wire, and therefore recorder may not be in this direction, it may be in another direction. Therefore, here in emission spectroscopy, what we are doing, in emission spectroscopy, sample is excited or it is excited by using suitable excitation energy. Therefore, this excitation energy is provided by the source. Excitation of atomic species or molecular species that will takes place at a specific wavelength, and therefore that particular wavelength we can obtain from the source by using a monochromator. Therefore, in emission spectroscopy there are 2 types of monochromator. One is a monochromator that isolate a excitation wavelength. That means this monochromator select a wavelength that is given to the sample. By that wavelength, this sample undergo electronic transition or excitation, and then here after some time sample emits radiation. This emission measured at the 90 degree to the incident beam. Therefore, here emission which is shown by the sample that is measured at 90 degree, and in order to select a particular emission line or emission intensity, here another monochromator is required. Therefore, here this monochromator gives a radiation only that is emitted by the sample to the detector, where it is converted into electrical energy, and that electrical energy is given to the recorder. Now here this is for the molecular absorption or this may be for a molecular emission spectroscopy as well as for a atomic emission spectroscopy. More specifically, we can say that, suppose for example, in flame emission spectroscopy, flame emission, that is a emission spectroscopy. In flame emission spectroscopy, here sample is excited by using thermal energy, and therefore here little bit different arrangement is made. Here sample which is given on the flame. Therefore, flame itself acts as a source of radiation. Then here a monochromator. Then monochromator to detector. A detector to recorder. Therefore, in flame emission spectroscopy, no doubt it is a emission spectroscopy, but in this case, the sample is excited by using thermal energy. Therefore, sample is aspirated into the flame. After that, atomization of sample species takes place and atom undergo electronic transition. Then that emits a radiation. Those are isolated by this monochromatic system. And here this detector converts that radiant energy into electrical energy. This is a arrangement of the component in flame emission spectroscopy. We are also familiar about the atomic absorption spectroscopy. In atomic absorption spectroscopy, again there is sample. Sample is given on the flame. Here flame acts as a sample cell. It acts as a sample cell. There is another source, and here this source provides a radiant energy for the or to the atoms present in the flame, and after that this flame, atoms in the flame that undergo the excitation by absorption of this radiant energy, a radiant power of this beam is decreased. That is measured in atomic absorption spectroscopy. In molecular absorption spectroscopy, radiant power measured that is absorbed by the molecular species. In atomic absorption spectroscopy, radiant power is measured that is absorbed by atomic species. Therefore, in both instruments, decrease in radiant power is measured. Again, additional here source that is placed in front of the sample cell. Here flame is a sample cell. The purpose of flame is to atomize the sample as well as to hold the atomic species into the flame, and therefore here radiation source excites this, gives a, again here there is monochromator to isolate a specific line which is given by the source. That emission line, its intensity is detected by detector, and after that it is recorded. Therefore here the instrument which is used in atomic absorption spectroscopy have arrangement like this. Therefore here we have to furthermore we have to discuss flame emission spectroscopy as well as atomic absorption spectroscopy. In that case, the arrangement of the component is in this fashion for atomic absorption spectroscopy. Student, now we discuss the instrumentation which is used in molecular absorption spectroscopy. The instrument which is used in molecular absorption spectroscopy are of two types. One is called as colorimeter and second is called as spectrophotometer. This classification is based on the type of region, that is electromagnetic region, utilized for the chemical analysis. Therefore, instrument for a absorption spectroscopy are colorimeter and the absorption spectroscopy or spectrophotometry are colorimeter when it is measurement is carried out in visible region, particularly from a 400 to 750 nanometer, or sometime 380 to 750 nanometer, and sometime another instrument is called as spectrophotometer, where the wavelength of analysis is ultraviolet and visible. Therefore, here this instrument works from 180 nanometer to sometime 750 nanometer, or in modern instrument it can be measured up to 1100 nanometer. Therefore, here similar to the optical instruments, here in this case there is number one is a radiation source. From radiation source to monochromator, to a sample cell to detector, and detector to recorder. This is a arrangement that is also found in colorimeter as well as spectrophotometer, because these are the instruments of absorption spectroscopy. Here for colorimeter, the radiation source is tungsten filament lamp. Tungsten filament lamp, that is lamp similar to our household lamp. Then monochromator, that may be sometime filters, or in few cases prism may be used. Then here sample cell are the glass tubes or glass cell. Detector is a photo cell or photo cell or photo tube, and record. And here this is a analogous recorder. These are the components of the colorimeter, that is a radiation source is tungsten filament lamp, monochromator are filters, sample cell that may be made up of glass tube, transparent glass tube, generally soda lime glass are used, detector is photo cell or photo tube, recorder is galvanometer. The components which are required for spectrophotometer are number one, here again tungsten lamp plus a deuterium lamp, that is D2. Then filters or monochromators are prism, or sometime a diffracting grating can be used as a monochromator. Here spectrophotometry works in visible and ultraviolet region, while colorimetry works only in visible region. Therefore, in order to carry out the measurement in ultraviolet region, the glass is not allowed as a sample cell. Therefore, here a quad cell or a cuvette that can be used as a sample cell, and here the detector is a photomultiplier tube. This photomultiplier tube is more sensitive than photo cell and photo tube, because here very weak intensity of light that can be measured by this photomultiplier tube. Here recorder that may be sometime a computerized system or computerized system or sometime a digital display or electronic circuit, that is a recorder. Therefore, here colorimeter works in visible region 380 nanometer to 750 nanometer. These radiations are visible. That means we can view this radiation. But radiation from 180 to 380 nanometer, that are ultraviolet, we does not observe that radiation. And this ultraviolet region to visible region, here a spectrophotometer can measure the degrees in radiant power of the sample. Here in modern instrument, this measurement may be carried out up to 1100 nanometer. But in this case, there is a infrared region. Therefore, the absorption does not possible in this region. There is a vibrational transition or rotational transition that may be occurs at this longer wavelength. Therefore, here this is the arrangement of the absorption instrument, that is a radiation source, monochromator, sample cell, detector and recorder. For colorimeter, we need a radiation source, tungsten filament lamp, filters as a monochromator, glass tube as a sample cell, phototube or photovoltaic cell that can be used as a detector, and galvanometer can be used as a recorder. While in spectrophotometer we require ultraviolet region, that obtained by using Deuterium lamp, visible region by using tungsten lamp, prism or a diffracting grating that gives a very narrow bandwidth of a monochromatic radiation that can be obtained here. Here quartz is used because that is transparent in ultraviolet region, while glass is not transparent in ultraviolet region. And photomultiplier tube, that is a sensitive detector, is used as a detector in spectrophotometer, and sometimes computer may be used as a recorder. Now we discuss the first component of the instrumentation, that is a radiation source. Therefore, first component is a radiation source. We know that for visible region, radiation source is a tungsten filament lamp, and for spectrophotometer there is requirement of tungsten lamp as well as the ultraviolet deuterium lamp. Here, if we consider the radiation source in spectroscopy, there are two types of radiation source. First type is called as continuum source, that gives the all possible wavelength in a required region, that gives the continuous spectrum of electromagnetic radiation, gives continuous spectrum of electromagnetic radiation. That source is called as continuum source. A source which is used in colorimeter or spectrophotometer, this is a continuum source. Tungsten lamp, deuterium lamp, these are the continuum source. Second type of source that is used in spectroscopy is called as a line source. Line source that gives only specific lines of the from the source, lines from this source, and this is called as line source. Line source is generally utilized in atomic absorption spectroscopy, and that is a halocathode lamp. Halocathode lamp is a line source used in atomic absorption spectroscopy. Therefore, source in spectroscopy is classified to two types: continuum source that gives the all possible radiation within a spectrum of electromagnetic radiation, and that gives a continuum radiation. Means here 180, 181, 182, 183, even down 183.5 like this. That means here continuous radiations are provided in a given electromagnetic spectrum by a continuum source. Here only specific lines are given. That means if you consider the source as a halocathode lamp, that gives a line like 280 nanometer or sometime 350 nanometer in this case. Only these two lines are generally given by this halocathode lamp, and therefore it is called as line source. Line source is more specific than continuum source. Therefore here these lines are the identity of the material. Therefore here AS is a interference free measurement. Therefore here we discuss the radiation source or used in colorimeter or spectrophotometer. First is called as a tungsten lamp. Tungsten lamp, that is a W lamp. This source is similar to our household lamp. Here this is a tungsten filament which is placed here, a tungsten filament. Here this is a glass bulb. Inside the glass there is argon gas, that is the inert medium, and here this is the power supply. This tungsten lamp operated at temperature T is equal to 2900 degree Kelvin, and here in this case it gives the wavelength from 350 nanometer to a 2200 nanometer. Therefore here radiation source which is used in colorimeter is a tungsten filament lamp. It gives radiation from 350 nanometer to 2200 nanometer, but visible region is only from 380 nanometer to 750 nanometer. Therefore other wavelengths are not required for the measurement. Now how does this lamp is operated? In this glass bulb a argon gas is filled, that gives the inert atmosphere, and here this is a electrically heated tungsten filament. Tungsten have very high resistance. Then when we apply electric field, this filament becomes red hot, and because of this that gives this radiation in this region. Therefore this radiation 350 nanometer to 2200 nanometer, this possible range of radiation are given by this tungsten lamp. Therefore there is again some advancement done in this tungsten filament lamp, that is called as tungsten halogen lamp. Tungsten halogen lamp. In this case a provision is made to add a iodine, and therefore here this lamp is little bit different than this household lamp. This lamp is like this. Here this is a coiled window. Inside the window there is again argon gas. Or inside this is a glass. Again this is a tungsten filament, and here a little amount of iodine here I2 is added. Here this lamp operated at temperature T is equal to 3200 degree Kelvin, 3500 degree Kelvin, and it gives wavelength from 240 nanometer to sometime 2500 nanometer. Now here this lamp is called as tungsten halogen lamp, which is a little bit derivative of tungsten lamp. In this case iodine are added. Here similar to earlier case, here this tungsten filament is heated, it becomes red hot and it emits a radiation. But while the tungsten filament is heated, at that time here the tungsten filament is get vaporized to some extent. Therefore, sublimation of this tungsten filament takes place, and because of this, if iodine is present, then Wi2, a compound, is formed in the inside this chamber, and this again diffuses back to this heated filament, and that increases the life of the tungsten lamp. Therefore, here life of this tungsten lamp is more, tungsten filament is increased because of addition of this iodine, and another advantage is that we can also extend the wavelength from 250 to 2500 nanometer with the addition of iodine. Therefore, tungsten lamp and tungsten halogen lamp generally use is spectroscopic instrument. Generally in colorimeter tungsten lamp is used, but in spectrophotometer tungsten halogen lamp is employed because that gives the wavelength of 240 nanometer to 2500 nanometer. Now second radiation source which is used in spectrophotometry, that is called as deuterium lamp. Deuterium lamp or D2 lamp. Deuterium, that is a heavy hydrogen. Therefore, here deuterium lamp is constructed in similarly. Here this is a glass bulb. Again there is a plate which is connected electrically. This is a heating plate consisting of platinum rhodium alloy, that is a heating plate. Here electric power supply is connected. Inside this chamber there is D2 gas of low pressure, and this is again a quad window. Quad is required to pass the ultraviolet radiation because glass cannot be transmits UV radiation. And this is the construction of deuterium lamp. This deuterium lamp that gives the wavelength from a 160 nanometer to 380 nanometer. 160 to 380 nanometer, this wavelength is given by this deuterium lamp. And here, how does this wavelength is generated? In this case here, this filament, it is electrically heated. Therefore, because of electrically heating this rhodium alloy, platinum rhodium alloy, this D2 gas undergo excitation, and when it get converted to two deuterium atoms, then a photon of H nu, photons are emitted in ultraviolet region, and that gives their 160 to 380 nanometer radiation. Therefore, here hydrogen This may be hydrogen H2, that gives H2 excited, that gives H plus HU, and this HU is energy. This is in the form of photon, and this energy is in ultraviolet region, having wavelength from 160 nanometer to 380 nanometer. This have a maximum band at 250 225 nanometer. Therefore, when in a spectrophotometer there is tungsten lamp as well as deuterium lamp, at the instrument shifted from UV lamp to visible lamp at this wavelength, that is at 350 nanometer. Student, this is about the radiation source used in spectroscopy, that is spectrophotometry as well as in colorimetry. With this, we stop here. Next time we discuss the wavelength selector used in spectrophotometry, types of wavelength selector. Then we discuss sample set and further instrumentation. Thanks for joining me. Wish you all the best. Keep learning. Happy learning.